The overall goal of this procedure is to sort mycelial pellets formed by filamentous Streptomyces according to size using a large particle Copus flow cytometer. This is accomplished first by the growth and subsequent sampling of the desired bacterial strain. In the second step, the sample strain is measured by Copus. The data is then analyzed in silico, and the mycelial pellets are sorted according to user-defined parameters. Ultimately, these analyses can be used to further characterize the pellet size heterogeneity.
Though this method can be used for Streptomyces, it can also be applied to other organisms such as filamentous fungi. Demonstrating this procedure will be Marloes Petrus, a PhD student from our lab.
First, grow the cultures by adding 100 milliliters of yeast extract malt extract medium and 0.2 milliliters of 2.5 molar magnesium chloride to one sterile 250 milliliter Erlenmeyer flask equipped with metal coiled springs for each bacterial sample. Then inoculate each flask with 1 × 10 to the 8 spores of Streptomyces to obtain a spore concentration of 1 × 10 to the 6 spores per milliliter, and grow the bacteria at 30 degrees Celsius with shaking at 180 rpm. After 2 days, use a sterile 5 milliliter pipette to collect a 5 milliliter sample from each culture while gently shaking the flask to evenly distribute the cells. Then dispense each sample into individual 15 milliliter conical tubes. To evaluate the samples by Copus analysis, confirm that the Copus Plus, pump, computer, and 400